Sure. So I'm a clinician scientist here at CGEn, and a pediatric oncologist is my clinical area specialty. And my research focuses on cancer genetics, particularly two areas. One is, I guess, the longest standing area has been a focus on cancer predisposition, and particularly the role of genetic alterations in predisposing children, young adults, or anybody, I guess, in the context of cancer predisposition syndromes. And we focused our studies, which are both basically very fundamental, basic genetics, genomics, biology, as well as clinical studies in a relatively uncommon cancer disorder called Li-Fraumeni syndrome, where children, basically, it's a lifelong disease in the sense that one's at risk for developing a wide variety of cancers. And the gene that is the driver is p53, a classic tumor suppressor gene, but we know that there must be many other alterations at a genomic or genetic or epigenetic level which modify the phenotype. So we've been exploring this in quite a number of ways that we can chat about. And then at the clinical level, basically, one of the oncologists here at CGEn is taking care of kids with a variety of cancers. I tend to focus on the non-brain solid tumor patients. So that's, I guess, what I do.
Great, thank you so much. So can you tell us something new or surprising that you've learned lately in the work that you're doing?
So I guess a couple of things, one of which we've just published last week in JAM Oncology, and another which data that's still unpublished but very exciting. So the published work is interesting, and I think from a genetics landscape, maybe not so much, a little bit genomics, but definitely genetics, is the concept of syndrome. So the idea being that there are a number of syndromes out there, and they classically are defined by a constellation of alterations or congenital alterations in an individual, which can be traced back to usually a single gene defect of some sort. And so the term syndrome has kind of been out there for a long time. So Li-Fraumeni syndrome, the one that I study, is a constellation of cancers which can occur at any age. Some people get one, some people get multiple tumors. Some first tumor is when they're a baby, their first tumor might be when they're 40 years old. So it's a constellation of tumors, but we now, in analyzing the families, a large, over 1,000, actually it was over 3,000 families, as well as the p53 database, so looking at where the mutations occur in the gene and can you correlate them with the phenotypes, we actually are redefining the syndrome as a spectrum. So in other words, one lies somewhere along the spectrum, depending on how penetrant the gene defect is and how what the phenotype looks like. So a very, very classical to a much less clear. And so by doing that, you might ask, well, what's the value of doing that? So I think that there's two. One is that it gives us a much better handle on being able to predict, based on a genetic alteration, what phenotype an individual may have. And if you know that from a clinical perspective, you can think of ways to try to predict more accurately what tumors they'll develop and at what age they'll develop it, and then you can sort of clinically manage them accordingly. So then the second part is it sort of leads to this idea that there are a whole bunch of other cancer syndromes, which really aren't syndromes. They're really a spectrum of disease that is dependent on more than just the single gene alteration, the driver mutation that we talk about. So that was quite exciting, and it's gotten some interest in the cancer genetics field. And then the second area, there's several we're quite excited about, but you asked me for two, so I'll give you two, is this taking further this ability to predict. We're not at the point yet where we know how to prevent these patients from developing cancer. So the idea is, can we actually predict whether they will? And if so, you could intervene, sooner and pick them up earlier. So using large-scale methylome sequencing, genome sequencing, as well as artificial intelligence machine learning tools, we're coming up with approaches using all of those together to more accurately predict both age of onset and perhaps ultimately tumor type in these patients. So this is exciting work that we're working with Anna Goldenberg and others to try to hone down, and we'll see how that plays out over time.
That's very interesting. Congratulations on your publications. Very, very exciting. Just a follow-up question. How big is your studies? Like, are these SickKids patients? Are they across Canada?
So yeah, so the patient numbers within SickKids, I mean, this is one of those rare syndromes. So within one institution, there simply aren't enough to really do those kinds of studies. So we actually, yes, do have collaborate with colleagues across the country, and frankly, more effectively, more importantly, rather, across the world. So in both of the studies that I cited, but in really most of the work that we do, we have a library of patients recruited from around the world. Yeah.
Thank you. So what technology, process, or other applications, maybe in terms of this type of work, do you think it's important for other, either clinicians or researchers to be aware of that's been facilitating the research that you're doing?
So, traditionally, up until the technologies that are now available for deep sequencing, the traditional approach was generally single gene or small panels, and in the cancer world that's been the case for some time, and in a sense it's been a little bit ahead of the game, just because there are genes to look for that we know that they do. So we, as with many others, would basically do single gene, i.e. p53 sequencing on these families, identify a mutation, and then kind of take it from there. What we've now done is take it to the next level. So we're doing whole genome sequencing, we're doing RNA-seq on tumors, as well as now methylome sequencing. And with that, what we're much better able to do now is refine the genetic landscape of both the tumors that occur in these patients, as well as the germline normal, if you will, to get a better understanding of, in addition to the driver mutation in p53, what other genetic alterations are occurring that are modifying the phenotype. So what we anticipate in short order is that, and this is actually already kind of happening, is when a family or a patient presents with clinical features of the syndrome, sort of a cancer history of some sort, rather than just doing p53 sequencing, the more typical way that we're now doing it is they actually get a large panel sequencing or whole exome, and we're doing that now through the SickKids Cancer Sequencing Program, which is also supported through the CGEn network. So that is that's a huge advantage, because we're not just finding a mutation in p53, we're now being able to find what else is going on in their genome. So those are the technologies that I think we're now using, and then gradually rolling them out for broader use.
That's incredible. That's very exciting. So how long approximately has that new change to whole genome sequencing been going on for now?
So I would say that we set up the SickKids Cancer Sequencing Program here was set up just under 5 years ago. And from the beginning, we were doing RNA sequencing, whole genome, and a large panel sequencing, which was created by my colleague Adam Schlein, who oversees the cancer genomics bioinformatics core of that program. So I would say it's about 4 to 5 years. What I think is really remarkable with respect to that is that initially it would take us many weeks, if not months, for results, for the turnaround time for results. So they weren't clinically as relevant because 6 months is a long period of time. We now have that time frame, at least for the panel, down to about 2 weeks, which is clinically relevant. That's a very practical term. The whole genome sequencing is still quite a bit longer, and that's not so much a technical challenge. It's more resourcing. The bioinformatics, as you know, for genome sequencing is more complex.
That's great. And would you briefly explain the difference, maybe just for those people who are not, as familiar, the differences between those three? So the whole genome sequencing, the RNA sequencing, and the panel. If you could just briefly, identify what the main differences are between them, I think that'd be interesting.
Sure. So when we talk about a panel sequencing, what we do, whether it's in cancer or in heart disease or in lung disease or you name it, what we're doing there is selecting a number of genes, and it will vary from disease to disease or indication to indication. But all those genes have been shown in one way or another to be associated with that disease. So in our case, these would all be so-called cancer genes, and genes that are involved in some way or another in cancer formation. And those, there's a large number of those. In ballpark, there's at least 1,000. There's probably many more, but that's what our panel is about. So then what you're doing, you're basically, what I would, you could think of it as if you had an extremely large, let's say, encyclopedia, let's say the Oxford English Dictionary, that's maybe a better one. Okay, so a panel, you could imagine that every chapter of the Oxford English Dictionary, every part of it would be one of those genes on the panel. Okay, so you have a pretty good idea where you're going, but there's still a lot going on underneath. The exome, the whole exome sequencing, is more detailed. So there, what you're looking at is what we call the coding region of the genes. So those are the parts, if you will, the words that make up the sentences of our genome. And there, you're able to actually sequence many thousands, because there are many thousands of genes. There's 25, 30,000 genes in the human genome. So you're basically mapping out all those 25, 30,000 words. Whole genome sequencing is further down. There, you're actually looking at the letters of the words. So there we have 3 billion letters, actually 6 billion, because there's 3 billion on each strand of DNA. So you're looking at every one of those letters, which make up the words of the exome, which make up the panel. So that would be, I guess, one way to think of it.
Wow, thank you for explaining that.
One thing, so one thing about the genomes is, so if you think of the exomes being the words, there's spaces between words. The genome is sequencing through the spaces, and we know that there's information in those spaces that the exomes wouldn't be able to translate, but the genomes would.
Great. Thank you. So what do you feel is the most exciting thing about genomics right now? I know there's a lot, but what would you identify as your most, to you personally in your work, what do you think is the most exciting right now?
Yeah. I think what's very exciting is that, well, a couple of things. First of all, that this sounds very practical, but it's important. The cost and the analytic technologies have dramatically improved. The cost has come down dramatically, and the analytic abilities have improved dramatically over the last few years, and constantly are being. And there's constantly new algorithms and things to be more, get a better understanding of what we're sequencing. So this is exciting, because it wasn't that long ago when the cost was prohibitive, and it was only in a very few number of places you can actually do this stuff. But now we start, this last week I was at an international meeting virtually with a number of people from low and middle income countries, and we can actually start thinking and talking about ways that these technologies could be utilized in countries that previously would have no capacity to do this. So this is exciting and very important, because we don't want to be creating technologies that are only able to be used in Toronto, Montreal, and Vancouver, right? So this is important. From a science perspective, these technologies are really allowing us to get a much better, constantly better understanding of who we are and how we're made, and why, how genomes are make each of us who we are. And I think a very practical and real example that everybody can relate to is the COVID story. It was really through genome technology, this incredible ability to sequence the virus within days of its first discovery, and then being able to share that data immediately with other centers who had expertise in creating RNA vaccines and so on and so forth. So that couldn't have possibly been done not that long ago. We would still for sure be waiting for a vaccine. So I think those are sort of two of the very exciting things. It's the very rapid translation into clinical practice that I think will continue to move forward. One thing to add in the cancer field is that this ability to sequence the genomes of tumors has very rapidly led to an ability to translate that information, identify molecular changes at a very precise way, to then match those to the development of drugs that target those particular molecular alterations. And in the old days, not so long ago, you'd have to sequence one gene at a time, and you'd have to have an idea of what genes you wanted to sequence, because you can only do so many. So you're very limited. Now we can go in at what in a sense, we call it agnostic, where we're not expecting anything. We don't know what we're going to find, because we can sequence everything. And then from that, we're able to garner out the information that's most relevant. And then new drugs are being designed targeted against those genetic alterations.
That's amazing. Thank you. And so in terms of the future then, since there's just so much rapid progression right now, what do you see for the future of genomics? Where do you think it's going to go from here?
Yeah. So I think it'll continue to build. The technologies will continue to improve. We now talk about doing long-range PCR, so being able to do large fragments and in a rapid period of time. We are talking about developing even more rapid turnaround time to make this technology clinically relevant. We're talking about smaller or panels that are very precise, so that you can use devices that are not taking up oodles of space, physical space. Advances in cloud computing are allowing much more ready access for data sharing, which is critically important in all of this science. Very few places have enough data, even with all the sequencing, to really dive deep into disease analytics. So I think all of those sort of technological advances will just speed up our ability to do better. I think one of the challenges going forward is, in a sense, the ability for regulatory and other bodies to incorporate genome sequencing in its various forms as a clinical test, as a clinically util test, in which case government funds the sequencing. And in Canada, that's not happening as yet, but in other jurisdictions in the world, it definitely is. And one of the ones recently that I'm learning through colleagues is in France, where basically cancer and other disease genome, whole exome, and relevant panel sequencing and RNA sequencing is now funded entirely through the federal government on all cancer patients, pretty much. So we know this can happen, and there's all sorts of reasons why different jurisdictions take longer to get there, but that will be one of the big pluses down the road, and I think the relatively near future, to be honest.
And I guess you personally, like as an oncologist, how do you see that as extremely important for the outcome of your patients at this point in time?
So we, through the KICS program, the cancer sequencing program I mentioned at SickKids, and then we have a national program called Profile, Precision Oncology for Young People. The sequencing, so the purpose of those is set up is that we're doing sequencing of patients with hard to cure cancers who it can have tumor sequence, and then from that we identify molecular targets that we then work to get drugs for, targeted drugs or immunotherapy as an example. So without the genome sequencing, you would have no way to know which drugs to use. And without the drugs, the genome sequencing would be of relatively little other than academic value. So the combination of these two has completely changed our ability to care for these kids. And we know even in the 4 to 5 years that these programs have been up in Canada, and there are similar programs around the world, that children and young adults who had basically no hope for cure or no hope for effective treatment now do. Not all of them, but the actionable targets that we're finding, the ability to match to drugs or immunotherapies continues to increase. So I have patients in my practice who have clearly benefited, that we've run out of typical traditional options, and then we find something, they're put on a new drug, and you're able to achieve remissions and so on. So it's incredibly important. It's absolutely incredible. I mean, we're basically, it sounds a little trite, but we're changing lives. And the genome revolution and the ability to sequence large-scale sequencing across the country for every single kid in the country who is eligible. These are national programs that would never have been possible without this.
Incredible. Is there anything else that you would like to add or touch on in terms of your work, in terms of CGEn, or anything else that we haven't covered?
I think in terms of the work, I mean, I could go on forever about the work, but I won't. I think in terms of CGEn, I think one of the things that's really valuable, we've talked about, different projects and how different technologies are used for things. And really, a lot of, it's so dependent on the concept of CGEn that you have a national, sort of a multi-institutional effort going on in terms of the foundational structure for these programs, because the amount of data that's generated is huge. So there needs to be a way to tie it all together. But at the same time, at an individual level, what we're finding with all the sequencing is that every child in our case is different and has these nuanced differences, which are very hard to tease out at a single individual basis. So the only way to do that is by pulling this data together in large numbers, like through the network, and then being able to take that information and then kind of turn it around to the individual patient in front of you and say, okay, we can now kind of put that patient's genome information, and they kind of fit into one or another spot based on the large scale sequencing. So I think that in my mind, yes, we could do sequencing without CGEn. I mean, I think it's a technology. It's fine. We can do that, but I don't think we would have the sort of capacity we do to really properly and effectively and fully interrogate all this information without it. So it's great. It's absolutely key.
Well, that's great. Thank you so much for sharing all these insights and about your work. It's really incredible. And we're excited to see where it goes from here. So thank you so much for taking the time to speak with me and to share all this with CGEn's community. And we'll definitely hear from you again soon, I'm sure.